Hi, good morning, everyone. It's time to talk about animals. So, what do you think if you are looking at this photo? That maybe they are cows enjoying a mud spa. But actually, they are Italian Mediterranean river buffaloes in their favorite environment. And of course, they are not just lying around all day, because they are part of a unique production system. And their milk is mainly used to make one of Italy's most iconic products, Mozzarella di Bufala Campana PDO cheese. But when it comes to production, mastitis remains one of the major challenges in buffalo farming because it's an inflammation of the mammary gland that affects both milk quality and yield. And in buffaloes, an early detection is difficult because we still don't have validated diagnostic tools. So we focused our attention on DNA methylation profiles in MIRB using nanopore technologies to identify potential epigenetic biomarker linked to mastitis. For these studies, peripheral blood and milk samples were collected from 100 female buffaloes, and we chose only animals that met these specific criteria. So we divided the animals into two groups, 8 as healthy and 7 as having mastitis. DNA was extracted from the whole blood with a commercial kit, and after libraries preparation, we sequenced it with the PromethION P2 solo. Base calling was done with Dorado, and then we aligned the reads against the reference genome of Bubalus bubalis. Then the methylation information was detected with the super accurate model and related data with modBAM to BET. Finally, the analyses were done in our software using the SS package. We checked our sequencing output with nanoplot, and in particular, Fred score and mean red length met all the thresholds, so finally all 15 animals were included in the analysis. We analyzed a consistent number of CPG, and in particular, DMC and DMR were considered significant based on a posterior probability, and we had an additional filter for FDR. We identified 22 DMC, and in particular, as we can see in this plot, a positive value mean higher methylation in healthy animals, and a negative values mean a higher methylation in mastitic ones. In particular, 68% of DMC were hypomethylated in healthy animals, and 32% displayed hypermethylation. In terms of DMR, we identified only one hypomethylated region located on chromosome 8. Genomic annotation showed different patterns. In particular, hypermethylated sites were mostly in the intronic regions, and hypomethylated sites were mostly in the distal intergenic regions. Also, we identify 8 differentially methylated genes, and in particular, 4 of these genes are involved in, have a predictor rule, and they are involved in balancing immune responses, managing cellular stress, and promoting tissue repair. For the other genes, we don't have a lot of information because they are still uncharacterized. So finally, this is the first study exploring DNA methylation patterns in buffaloes using nanopore sequencing. And also, even if the buffalo genome is not completely annotated, the technique and the pipeline we used showed a consistent, reliable result. In the future, of course, we will increase the sample size and integrate the RNA-seq to evaluate the gene expression, and could be useful to integrate this epigenetic data to improve herd health and dairy performance. I would like to thank my team in Naples and some special people from the NIA Institute. Thank you.